we'll go ahead and get started. Maybe some more people will wander in, we'll see. So I sent out a fix last night for those of you that were having issues with the R kernel. I believe that everybody that had an issue had it because they installed R Essentials before doing the rest of the thing, because I included that originally on the day one installs, and I had mentioned not to do it, but had accidentally had it in the documentation for you guys, so a lot of you did it. So for whatever reason, this program has a weird bug when I'm installing things. In this case, the order really matters. So I went through, the first command was just to remove the R library that we were working in, and then also uninstalling R altogether, and then just starting over fresh with the correct order of installation. So if anybody is still having problems with their R kernel, come talk to me about it after class or during class. So we finished DESeq2 yesterday and got through everything of differential expression, which is essentially the end of this pipeline. So my goals for today, we're going to do one quick thing with gene ontology. And then I'm going to also just give you guys a few random exercises that aren't necessarily linked to the processing of this data set, and introduce a couple other random tools that I find really useful but wasn't able to incorporate into the lesson in a logical place, which is why I haven't taught them so far. So today's going to be a little bit different. It's going to be a little bit more random and sporadic with what you're learning, but the goals of what I'm trying to do today is just expose you to a few other things that are out there, and now you guys should be experts enough to be able to latch on to just a little bit of exposure from what I've given you, and if it's something that you think you're going to use later on, be able to figure out how to do it on your own. So before we get into all of that, are there any questions, just general questions or specific to the code that we've done through these first, well actually through this entire workflow that anybody wants to talk about? Yeah?
What if we use a piece of software that cannot be installed yet?
Okay, we can get an example of that today. Yeah, that's another thing that I didn't have a chance to get to. Although, really, I have yet to come across something that I can't install in Anaconda. In teaching the new people in my lab, when I first started out, I didn't know about Bioconda, so I was trying to install a lot of stuff on my own, and it was really annoying. And somebody told me about Bioconda, and I was like, everything is in there, it's great. But I'll get to an example of that in class today. Any other questions? Cool. All right, so I want to start out with this because in the lesson that we did yesterday, we got through how to use DESeq2 and how to use the R kernel to analyze your data in DESeq2. But a lot of times, people will be asking you for an output file that you want to very simply be able to open in Excel. And our DESeq2 output has 20,000 rows, so that's not very useful, or it can crash Excel very easily. So this notebook is a workflow of how you would use Python to filter your differentially expressed genes that you get from DESeq2. I'm sure there's a way to do this in R as well, but I just don't know R, so I'm not teaching that here. But if you're more comfortable working in R, feel free to use Google as you need to, or other tutorials to figure out how to do this. But what this notebook is doing is it's taking the DESeq2 output, and I'm showing you guys a few simple commands that I use regularly to select specific rows of this notebook. So if we only want to output genes that have a significant p-adjusted value. If you want to use the entire file and send it to collaborators or look at it on your own with Excel, you could try to do that with this 20,000 rows. But if you want a way to kind of chop it up and make it smaller and more usable, this workflow here outlines how we're going to do that. So this is in the Google Drive. You guys can make your own Python notebook to start doing this workflow in. Remember, we made in our Jupyter home, we made a directory, in our home we made a directory called Python Notebooks. We also made a directory in there for Lin28B shRNA knockdown. So this is still using that data set, so it might make sense to put it in that folder and call it something meaningful. In this case, you'll notice the only Python package that I'm using is pandas. So that's my only import at the top. Pandas is a great Python library that essentially allows you to do a lot of data frame manipulation. So this is referred to as a data frame. And pandas is great to be able to do all sorts of things, multiplying across data frames, manipulating data frames, adding rows and columns, all that fun stuff. So you'll notice the syntax is the same as what we saw yesterday. We're going to define the directory where our dseek2 output is stored. And you'll remember we used dseek2 and R yesterday to save this output. And we're going to read it in. We're going to call it dseek2 output. And we're going to read it in with pandas.readcsv. And you remember to follow the correct structure with your directory information and your file name. And the rest of this should be the same so that you can see your output. Sammy, you need to send me the public like private key. Are you guys all able to reload up your notebooks? I realize that takes a few minutes, but hopefully the instructions we had from yesterday, you guys can follow those again. Are you guys having problems? It's just not doing it? Oh, that's fine. Yeah, that's just something that's just going to keep running while you're working. I just leave out on these things. You don't need it to be.
not running somewhere else. And you can check that, you can check that with the dash view username. And then if it's see Jupyter Notebook running there, you can kill dash 9 and then the number is basically like the process ID. And then it'll kill that, and then you can start please, But if you let you do the PS dash new space username, you can Jupyter Notebook it's not running, it's not running. Yeah, but you can see you can do it in the other screen first. Yeah.
Remember that if you're getting errors about a port is already in use or that it cannot do local forwarding and things like that, you can always check what you're running on TSCC with PS-U followed by your username. And if you're trying to load a Jupyter Notebook multiple times with the same port, you're going to get these errors. And so this is a good way that you can check to see what you're running. So if I were to look at mine, okay, these are all different things that I'm running at the time. So I've got a couple of things in QSub, some screens, we'll talk about those later, some logging commands, but no Python notebooks here. So if you have a bunch of things on here that say Jupyter and Python and you're not able to open it all and you want to clean your slate and start fresh, you bring up this page and you copy this PID number to kill it. So if you want to kill something that you're running, you're going to use kill -9 and then the number that's associated with it. I know I've mentioned this a couple times, but I don't think I actually wrote it for you guys anywhere in the instructions. So make sure you make note of that. It's a very useful thing to help you wipe your stuff clean. Huh?
You did write it.
Oh, I did?
Yeah, it's in the Jupyter.
Okay. It's in there somewhere. The other thing that a lot of you guys were wondering when you did your tunneling on your home computer, you're like, it's not doing anything, it looks like this. So when you're doing the tunnel, you're keeping the tunnel open throughout your entire Jupyter session because you're accessing TSCC, you're pulling your data from there, it's an open connection. So this command is going to be continuing to run while you're working on TSCC. Do you guys remember if I want my command line to come in, my typical workflow, I open a tab, you'll notice the only thing that's on this tab is this localhost command. So I open a tab and I tunnel my notebook and then I just leave this tab alone. I don't do any other work in here. And if I want to do other things on my desktop, I use a new tab. But if I wanted my command line to come back and I wanted this to be running in the background while I'm working, how do I have to modify the command? Do you remember what you can add to your command to be able to run it in the background? Got it? The and sign, great. So at the very end, if you add a space and sign, then this will say, okay, I'm going to run this in the background. You can think of it in the same way as you're like sending stuff away to TSCC. It's like, this is the command, go do it, and give me my command line back. you're still not able to get your notebooks up. Or do you have a question? Okay, we'll get to that if everybody has a notebook open. You're good? All right, let's keep going then. Okay, so we imported the package that we're going to use for this notebook. In this case, we're just going to use pandas. And I just want to point out, by convention, I always put my imports at the top. Doesn't matter where you put your imports. The only thing that matters is that you run the import command before you try to use the functions that are stored in that package. So you'll see right here, I'm using a function that's stored in this package by calling it after the shorthand that I've imported pandas as. So read CSV is a function of pandas. So I need to have this run before I'm trying to use the command. Sometimes I'll be working through a notebook and I'll be farther down and I'm like, oh, I want to use this other package to do this new thing. And I'll do it right there in the cell that I'm working on, import whatever as whatever. But I just have to, just keep in mind when you're working through these notebooks, it doesn't matter where you put anything, it just matters the order in which you run it. And you have to have something loaded into memory before you try to call functions on it. All right, so we have our imports. Any questions about this first cell? Everybody able to load up their differential expression matrix? Yeah. What? What's your error say? Are you sure it does? Did you check? So did you check the path? So you're getting it this directory? So copy that directory. And try to see into that. Okay. Let's look at it. Use tabs to replace. Yeah. Yeah. Okay. Okay, and now you can load it. You're on. Yep. Remember, you guys have to keep your logins the same when you're tunneling your notebooks. Remember, there's two different logins on TSCC, so you have to check when you load your notebook which login node you were on. In this case, I'm on login 1, but it will randomly change every time you log into TSCC unless you specify on your login. So I'm on login 1 over there, so I'm going to be in login 1 right here. Okay. All right, the second thing that we're doing, we're loading this file that I have made for you guys, where we're converting the GenCode gene ID, which is the output, into this gene name. That's handy. A lot of times we want to look something up by the gene name. We don't know gene IDs. So I've got this this file, in this folder. So you should be able to load it just fine. Just notice in here, if you're loading a data frame, in this case it didn't have a header, so I gave it a header name for that column, gene name. Okay, any problems loading that file? Okay, this next code, we're going to join that gene name onto this data frame, so now they're connected. And we have a new data frame. I'm going to assign it to a new variable called output with genes. And I'm going to add the gene name column onto this data frame so that we can use that to very quickly look up all the other values that we're interested in. Any questions about this cell? Okay, so here's where we're going to be doing a lot of the filtering, and this is where you can, make decisions as a user. Again, every time I make something different, we can check the shape and the shape of it with this dot shape. And now this is telling me I'm filtering down to about 1,200 genes, and this is a lot more manageable to attempt to open in Excel. And it's nice because all of these genes have a p-adjusted value of less than 0.05. So the way that you read this is that we're doing it on this data frame that we just made. So dseq2 output with genes is this data frame that we made. And we want to filter it by picking the rows that have a specific value with this p-adjusted. So the way that we do that here is with this .loc, and then we specify the column that we're going to filter on. So in this case, we're filtering on p-adjusted from this data frame, and we want values that are less than 0.05. And so we're going to take the output of that and assign it to a new variable. So now we have significant genes, which is a subset. It's only 1,200 of them. And now I can just save this file, and I can open that later on with Excel. Yeah?
So we're noticing this back row of all three of us that we all have different numbers of genes.
That are significant?
Different numbers of significant genes too.
Did you save? Okay, raise your hand if you have 1276 significant genes. 1275? 1275? Okay, so so it depends on where you loaded your file in from. So this is all from your differential expression. And when you started at the top, do you all have this number is different as well? Or this is all the same? It's different? Okay. So we pulled this from R and from dseq2. I don't think you guys pulled different. Did you all use the soft link BAMs to do feature counts? Because the counts matrix is what we inputted. And remember we were testing a couple different parameters with dseq2? I can't hear whoever said that. Speak up. And your counts matrices look the same on dseq2 when we started? No? Okay. Program from what?
We all use different numbers.
Use different numbers for what?
Like we ran a program instead of software boards.
Okay.
So ours are on that.
What numbers are you talking about?
Like when we wrote the code, like we
Yeah.
all put different numbers for like the maximum. Like you told us.
I see.
Yeah, it was
Okay.
over and over.
I see. Okay. Okay, that makes sense. Yeah, so depending on where you pulled your feature counts from and how you specifically wrote feature counts, you're going to get different results. And this is actually a really good point to bring things up. This is where you can see how the decisions that you make and how you're going to allow for mapping, how you're going to specify your counting parameters and things like that are going to give you slightly different results. So it's really important. Often it can be very tedious to go back through and change every single parameter and see how that affects your output and doing that over and over again. Depending on how, I guess, weird your results are looking, you can decide to go and debug certain things. But it's really important that you understand what parameters you chose so that you're interpreting your output properly. And a lot of times small differences are not going to make, they're not going to have a huge effect. But if, you're number of differentially expressed genes is significantly different, or when you go to validate the differential expression that you're calling, either on IGV or with qPCR in the lab, and it's not making sense, you need to go back and understand exactly the parameters that you chose when you ran the code, and maybe that could help explain why. Okay, good catch. Okay. So here, what I'm having you guys do is save just the significant genes to a CSV. And this is a much smaller data frame and easier to manage. And the reason that we're doing this is because we want to use these gene names to do gene ontology. So one simple way to do this is going to be to export this file off of TSCC, so using SCP to move it to your home, and then we can copy these lists of gene names and do gene ontology on that. Also, we're not using it here, but I wanted to put this code in here because this might be useful for some of you guys later on. If you have a specific gene in mind that you want to look up the value for within the massive data frame, you can also do it with this .loc. And here you can specify which column you want to look up and what value you want to find in that column. So here I'm looking up Lin28A, and I can see the stats specifically for Lin28A. Yeah?
So the fact that you don't really have a significant value, does that mean that the numbers
That what?
are knocked down to number?
This is Lin28A. What did we knock down? So when we go to Lin28B, mine's probably not running, so it's not going to let me do it. Oh, yes, it is. Very significant. And the log2 full change is pretty negative. Okay. So take your significant genes file and export it, or save it to a file, and use SCP to move it to your desktop. Actually, the Linux users might not have Excel. Do you have Excel? The other thing you can do, if you just want to import, or if you just want to save the index, so the gene ID, so you can do so here, I'm just taking the index of this data frame, so just this list of gene IDs, and I'm saving it to a variable called genes, and now I have this shape that's 1276 long and it only has one column. So if I look at that, I can see that just this list of gene names. So I could save that. The data structure is a little bit different, so I won't let you save it directly in the same way. But for now, you can just save the differentially expressed genes file and open that on your desktop. Even if the Linux users don't have Excel, go ahead and still do this to practice using SCP. You guys should remember the context to securely copy the file across from your server to TSCC. Or from your computer to TSCC. What? For SCP? No, you do. Source destination, source how it goes. And if you're not sure, how would you check it out? Yeah. You can open a new tab.
Source file. Yeah. And then you have to have like a cache.
If you forget how to use SCP, you can refer to the manual. In this case, what's our source file? And is that on TSCC or home? And what's our destination file? Stop. Okay, so we're trying to copy a command using SCP. When you use SCP, it's different than just copy. Why? Because we're transferring across the server. Okay, so when you're transferring across the server, how are you going to specify in your command what server it's located on? Where it's located? So with copy, we gave it just the full path of the files. With secure copy, you have to add something else to tell it what server it's located on. What else does that be to add? Okay. Right, so you're doing this to your login. So you're specifying that this file is on your login. And you're giving it your TSCC login. So is this a command that you're running from TSCC or that you're running from your home computer? Why are you running it from your home computer? Because you want to save it there. What else? You're exporting from TSCC. So when you're going across a server, you have to specify the location of the server that you're not on. So if you're on your home and you're trying to access a file in TSCC, you have to specify your login. You can do it either way. You can be on either server, but if you're on your TSCC, you have to specify login information to your home computer, and I don't even know what that is for mine. I just always know what my TSCC username is. So anytime I do SCP, I always do it to my home computer because I can very easily provide my TSCC login. So if I'm always on my home computer, the source or destination of where I put the login information changes depending on which direction I'm trying to go. So you guys told me that the source file is on TSCC, so I have to provide the login information, in this case, for the source file. I believe before we had a destination on TSCC, so I provided the login information with the destination file.
Our username at TSCC and then colon.
It's in the SCP file documentation that we did. So if you open back one of the notebooks, it's in number 3.
Okay.
So check back at the examples. Help you guys find it. Okay. I need to grab water really quickly.
Yeah, it's in step 3. And basically, I just use exactly the same. Yeah, the same idea, except with the path of what you want, the file you want. And that's totally right. And then keep the space dot slash, because that's where it's going to go, which is going to help. It ended up in mine. If you wanted a specific file in your computer, you can change that. It ended up being just in my own directory, but basically doing just that. So this is all the sources. The sources, yeah, that's all right. So change the, this is going to be your username and everything for TSCC. Yeah. Because our usernames are all UCSD.
Raise your hand if your file has been copied. Great. So you can open the copy of your file in Excel. And this is often what I'll do when I, need to send things to collaborators or whatnot. I'll send them a file that looks like this. So then you can, use something that you're maybe more familiar with and play around with stuff. But for our purposes, we're going to do gene ontology to see, well, who can tell me what gene ontology is? Is anybody familiar with this? Yeah.
So it's the one kind of units that was present. I think it's all the process of globalization. So basically, there are a bunch of almost every
Can you speak up a little bit? I don't know if everybody can hear you.
So basically, was there manually annotated standard annotated for function biological process and localization? You can perform clustering on these terms so that you can see, you can read certain numbers and you can basically infer biological data.
So we're trying to infer function from these big groups of genes. So one of the questions that you're going to be asked is, okay, all of these genes change in my system. So what? So maybe you have a couple targets in mind that you can specifically check. I think one of the best things to do is look at your most highly differentially expressed genes, see if they validate in your data set, and then read about them on PubMed and see if you can kind of identify any sort of pathways or any other clues that go along with that. But another way that you can do it is using these gene ontologies, which are these curated lists, manually curated lists, that group the gene name into what their known, quote unquote, known biological function is. I say this with quotes because you have to be very careful with gene ontology. Our categorization is not great, and like 99% of the time, the top hit is always cellular process. Cool. So I personally have yet to use gene ontology in my 2 years in the Leo lab. Yeah, cellular process is never one that you would report as something interesting in your data. But occasionally, you'll run across, and we've come across things in the lab where there seems to be like a over-enrichment for a bunch of stuff that's popping out about like the mitochondrial function. And that's something that we could then follow up with of a functional assay in the lab. So I want to point this out as something that you guys can do, something that everybody in the field will probably ask you to do. Did you run gene ontology on your data set? There's a bunch of different programs out there that do it, but we're just going to use whatever the first web kit is. Okay, so the way that we're going to do this is we're going to take our list of names, and depending on the source that you're using it from, they might take a list of gene IDs, which is here, or gene names. So we're going to specify the gene ID, so go ahead and copy that list. And then go to the internet and type in gene ontology. So there's a couple different ones. David is one, I think that is pretty good. We're just going to use the first one. And it says right here, for your enrichment analysis, your gene ID is here. So there's a couple different things that you could look at. You can look at within your, well, this is what we're going to do. So within our set of genes, I'm going to paste that list. Is there anything within that set that seems to be highly enriched? Another really important thing that you have to think about, given your experimental setup, is what your background could be. So instead of just looking for enrichment within a set of genes, you're looking for a significant enrichment over some sort of background. So maybe your background would be a list of non-differentially expressed genes in this case. Or because, it could be that you're just picking up like the most highly expressed genes, but not something that's actually relevant. So this is something where you can play around with, again, depending on your experimental setup. You'll hear people referring to background a lot, where you're supposed to give it some sort of rational, random list that is of like similar type to the list that you're looking at. But in our case, we're just looking at the list of genes that we've identified as differentially expressed and seeing if there's any sort of enrichment in cellular functions. So this is what Evan was referring to. You can do biological process, molecular function, or cellular component. So you guys can try a couple of these. And this is human, so we're good with Homo sapiens. So if you submit, so once it runs through, you'll get these lists that have a various associated p-value with them. So you can click on p-value to sort it based on the most significant gene sets that came up as enriched.
Did you do that?
Keep in mind, we talked about the corrected p-value yesterday, so depending on the program that they're using, they may or may not be recording the value as a corrected p-value. And here's our top list of hits. I was trying to turn this into something So David is the other website that people like to use. I believe K. K pathways.
So David does PEG pathways.
Okay. And when you're reading about papers in your field, you guys will see the lists that people are using and recording. There's also kind of a spin-off of gene ontology called gene set enrichment, or GSEA. This is another thing where they've developed, this has been developed, I believe, around the cancer field, where they've put like sets of genes in these packages that are known to be associated with a particular thing. And then they look through the list of genes that you've provided and see if any of your genes are falling, if a significant number of them are falling within the packages that they've built in their gene sets. And then they would say, you've got a lot of genes in your data set that are falling within this particular set. Yeah.
How do you define those sets?
Just based on experiments and what people have done, sequencing. If you sequenced 100, colon cancer patients and you see this set of consistently upregulated genes, that might be a gene set that's associated with colon cancer. or knockdowns or particular pathways. All of these sets in these ontologies are based on manually curated sets of genes. And a lot of times we'll learn new functions about genes, or it will be reported as like the first function that was found for that gene, but maybe it's not even its primary function. So again, you have to be very careful with these things. But they can be useful. Yeah?
So, is there ever be worth that when you do these analysis and also include like
To also do what?
like seeing like the actual, because you're basically just parsing through like a list of genes without necessarily considering like.
I see. So that's why we're using a metric of differential expression. So you could use like upregulated genes specifically or downregulated genes specifically. Yeah, if your question instead is not something that changes across your condition, so we're looking specifically at things that change upon knockdown, but maybe within this given system, maybe you're looking at a very specific type of tissue in mouse and you're interested in the gene expression within that own system, then you could maybe take the list of like top 10 highly expressed or 10% of highly expressed genes. In that case, you would consider expression. So it really depends on what it is that you're trying to ask and what sort of filtering level you're going to put on that. Yeah.
Where is it getting the fold enrichment information?
So the fold enrichment is relative to the expected. So these gene sets are all different sizes, and given the number of genes that are in your list, you would randomly have a chance of expecting so many of them to fall into each category. So they calculate this fold change based on how many of them are actually falling into the category relative to the randomness or to the random chance. That's how they're calculating the p-value as well. Yeah.
So how do you get to the which genes are within these functions? I'm like clicking on the GeoWell process, and that's just a description of what they're talking about. And then, so I'm kind of,
If you click on the number.
Yeah. So right here, this is telling you reference. So this is probably all of the genes that are in these sets, and number is the one of yours that are mapping to this set. So if you click on that number, here's all the mapped IDs that we provided, and then some more information about the families or the class that it came from. Again, these are all things that you guys can play around with on your own, but ultimately what's most important is that you can validate whatever result you're finding in your experimental system. And again, if you're overwhelmed by the number of tools that are out there and are looking for more of a specific place to start, I would recommend following the literature in your field and seeing what other high-profile papers in your field are publishing on. Okay. Any more questions on differential expression, alignment, counting, gene ontology? This is the end of our RNA-seq pipeline. We've made it. Good job, guys. You should now, in theory, be able to download the other 3 ENCODE data sets and fully process them, or whatever data set you want from the web. Be able to fully process it and determine differential expression on a protein that you're interested in, a setup that you're interested in, a disease and control system that you're interested in whatever context you want. Yeah?
so practically speaking, in my data set, I have over 1,500 genes that are coming out with molecular 0.05.
You have how many? 1,500. Okay. Yep.
So, practically, like, how do you go about then even beginning to look at that and see what actually want to pursue and actually
Right.
want to look
Right.
at
So,
on the scientific level?
yeah, so you've got 1,500, and that's really hard to put together in some sort of like manageable thing that I can understand. So if you want to try to do broad categorizations, start with gene ontology. What I also do is I look at the most highly upregulated and most highly downregulated genes. I look at the idea of what that actual gene is, and I type it into PubMed, and I see if maybe I can identify after searching through 5 to 10 to 20 genes, if I can identify anything common or interesting about them for my system. A lot of times I'll be looking at not just in the context of gene expression, but for example, we study RNA binding proteins, so we're studying how a binding protein, or how a particular RNA binding protein, when knocked down, changes gene expression of the cells. But we also have data about how a particular RNA, where RNA binding proteins bind and what transcripts they bind on. So then I could take the list of genes that were differentially expressed, and I could see maybe how many of those were also actually bound by that protein. So there's tons of different DNA binding, there's tons of different transcription factor binding and RNA binding protein binding data sets out there that you can mine, depending on your question. You know, maybe if one of the most differentially expressed genes that you see is a transcription factor, you could go to the ENCODE website or the genome browser, download the transcription factor binding profile for that particular factor, and see if the genes that are differentially expressed are also the genes that are bound in the promoter regions by that transcription factor. And that might give you a clue as to where to kind of guide your next experiments to identify whatever it is that you're working on. But
Yeah, I think this is just, you're asking a very basic question that a lot of biologists are asking. Right? Whenever they do a genome-wide experiment like this, you get lots of stuff. No matter what you do, like the chip, you know, we'll see whatever you get. These days, I think it makes more sense to overlap things, so integrate things together, like integrative analysis, try to test specific hypotheses that you have, right? Not just rely on one experiment. So if you think about it, that means that very often, before you even do an RNA experiment, like in our lab, we have it sort of set up that we have other things that will happen at the same time, right? Otherwise, there's no point in doing a large experiment, right? You plan several things at the same time so that you can integrate the data, try to make sense of it.
you will never approach your experiment to be like, your main question is just what gene, okay, sorry, you shouldn't approach your experiment having your main question be what genes change expression upon knockdown. You know, that question is embedded in your larger research question of how this protein is regulating, you know, development of cancer or a particular developmental phenotype or whatever it is that your question is interested in. So the RNA-seq is never like your only question, the end-all be-all to your experiment, but it's a very useful tool and can give you a lot of information on how to guide your experiments forward. Great question. Like splicing and stuff? No, not really. So, okay, so this is differential expression through RNA-seq. RNA-seq, you can also look at splicing, depending on what you're interested in. You can look at other features of the RNA. RNA has 5' UTRs and 3' UTRs, and the length of those things can affect the regulation. All of this is available in your sequencing data, and there's various programs out there that can let you analyze these other features of the RNA. So again, depending on what your question is, keep in mind, this is why we spent a lot of time in the beginning actually going over the theory of how all these libraries are generated and what it's actually, how we go from the cells to the sequencer and what the data is a representation of. Because if you have a really good understanding of that, then you can have a good understanding of what sort of questions you can actually answer with that data. You know, you need to understand where your data came from, what it's representing, to be able to understand if the conclusions that you're making are valid and if there are any other questions that you can ask. Yeah?
If you're working with a collaborator, what are the key things you want to make sure that you're matching up on with their data set?
So if you are both doing your sequencing experiments independently, chances are they're not going to be directly comparable, but as long as you have your own, you always have to have your own cases and controls within your data sets. If you're trying to have, you know, them do one mutation, you do another mutation, and you could both have a control, but you know that's generally not a good idea. If you're going to do an RNA-seq, the experiment itself needs to be together and complete as one entity. Again, making your libraries for all of your RNA samples together is very important so that you don't have batch effects. Sequencing on the same machine, not using a different Illumina machine or going from Illumina to PacBio, things like that. Even when you do sequencing runs on different days, you'll have small batch effects, but trying to keep everything as together as possible is great. But ultimately, if you're sharing data and comparing data, assuming that within each experiment you have it set up well, where you have your cases and your controls together, things like genome builds, if you're trying to do direct comparisons, species, making sure that the sequencing was done on the same machine and things like that can be useful.
That's again a very broad question on design of experiments, right? So you actually do a comprehensive experiment, you think about also biological and clinical variation, right? And I think for collaborators, they should understand, make sure that I would say the collaborators that understand that. Right?
Yeah.
Not all biology.
Yeah, if you're trying to do a sequencing experiment, with a collaborator, you're generally doing it because either you have the system and they have the computational tools, or they have the system and you have the computational tools. But just, work together beforehand to kind of set up something that makes sense. But now you all have the computational tools, so you can rely on yourselves. Okay, great. So that completes the RNA-seq analysis. Again, you guys will have access to the Google Drive with all of those things. I'm not planning on taking those notebooks down, but I would recommend if it's something you think you're going to be useful for you later on, save them to your computer so you have these resources. Also, we have these videos that I've been uploading to YouTube, so I will send you all the links to those at the end of class as soon as I finish putting them all together. And if there's anything that you want to refer back to, feel free to do so. Also, if you know of anybody else that's asking you to teach them these things because they're like, oh, this is so cool, I want to learn it too. You guys feel free to share all the resources with whoever you want, and the videos as well. All right, I'm gonna go a little bit in a different direction and just kind of show you guys some other things that I find useful that we didn't have a chance really to get to in class. So we talked about submitting jobs to TSCC. The other thing that I do actually every day is I submit an interactive job. So this is different when we're then writing the PBS script, where we're telling it what sort of processors and nodes we want, sending it away, having it do our process, and sending it back to me. That can be really inefficient if you're still trying things out with your code, if you're not sure if it's exactly how you want it, you want to see the outputs as they're coming, you're debugging things, you're testing things along the way, but you still really need that processing. It's too much to be running it on the head node. The way that you can do that is to submit an interactive job. So this is exactly the same as submitting jobs, except it's done in an interactive mode, meaning all of the processors and nodes and wall time that you've reserved is actually now being run on your head node. And you do this with the Q sub -I. So again, you specify the you specify the queue that you're submitting to, you specify the amount of processing that you want, and you specify the wall time. And now your head node, it will have you'll see a different login. It'll have a job ID name now, and your head node will be running with this amount of processing. So you can be doing those higher memory intensive jobs, and you can do it in an interactive way. The one thing that's different about this, so remember when we were submitting jobs, your wall time, whenever your job was done, it just stopped. Here, your wall time is what defines your interactive job. So your interactive job will be running for the exact length of time that you specify, unless you kill it early. So these can be very useful. This can also be useful when you're running your Jupyter notebooks. If you're trying to do something in the notebooks that's very memory intensive, you can first start an interactive job and then load your notebook in that interactive job, and it will be running off of a higher processing power. So if you're doing some stuff in your notebooks and you're getting this error, it's like memory error, we're dying. Not just dying, but actually a memory error. The kernel error that we saw yesterday where the kernel was just dying, that wasn't a memory error, it will say memory error. And then you could try to go with this approach. The other thing that I find is really useful is using screens. So I've included a link here where you can read more about how screens work. But essentially what they are is you'll notice anytime you guys were done for the day, you logged out of TSCC, you lost your internet connection, anything that you were running would stop running because you've lost your connection. But what you can do is you can go into a screen before you start, and it's something that kind of runs in the background, and even after you log out of TSCC, it will still be running. So if you're, like for example, some of you guys were running feature counts on the head node, but it took maybe 10 or 15 minutes to run. If you were to run that in a screen, then you can get it started, walk away, and not have to worry about losing your internet connection. So the syntax for these is a little bit funny. Why don't we actually try an example of one of these together, just because the commands are kind of weird. So to start a screen session, you do screen-s, and you can specify the name of the screen. This can be useful because sometimes if I'm working on multiple projects at a time, I'll have a specific screen for each project that I'm working on, just as a way to keep myself organized. And it's useful if you have a screen name associated with each one. So you can call it whatever you want. It's going to take you into a new window that's going to look exactly the same as the terminal window that you're in, but you're going to be in a screen. So sometimes it can be hard to remember if you're in a screen because it looks exactly the same, but keep track of what you do. Once you're inside of the screen, you can get out of it with Control-AD. This is like a keyboard shortcut to leave the screen and go back to your head node. When you're on your head node, if you're not sure all the screens that are running, you can do screen-ls to re-enter a specific screen. So you've logged out and now you're logging back in and you want to go back into that screen where you had something running while you were done. You use -R followed by your screen name. And then to kill them, you can either type exit in the screen, or you can type -R, or sorry, you can specify the name of that screen and use -X quit. So screens are a very common thing. If you Google how to use a screen in Bash or on a remote server or something like that, you'll see all of these commands explained to you. Yeah?
Can you say again what a person will use a screen for? It's just having a working space or something?
The screen allows you to continue letting your jobs run while you don't have an internet connection or if you lose TSCC. So there's some things that I will run that take a really long time, and it's like something that's submitting multiple jobs while it's running, but I need to have the command line stay up while it's going. But if I lose my internet connection, then I'm going to stop the job and it's going to be really annoying. So if I first start a screen and then I run that command in the screen, it's going to continue running no matter what. So I can close my computer and it will still be running. It doesn't matter if I lose my internet connection. So if you wanted to start your feature counts job before you're done with class, but you have to go, you can enter a screen, type feature counts to start it running, know that it's going to take 15-20 minutes, but it doesn't matter. I'm going to exit, log out of TSCC, and when you come back, it will be finished for you. It won't stop. Yeah. So Q sub is sending your job away remotely to TSCC. You can't touch it. Interactive job, you're getting the memory, you're touching it on your command line. Screen is nothing to do with submitting jobs. You're just operating on your head node. It's just like basic command line, but it can run no matter what if you lose your internet connection or not. More questions?
Is that true if you have like a Jupyter notebook that's facing your screen, I guess? Like even if you lose your internet connection?
The notebooks don't exactly work with screens because of your tunneling, and when you lose the internet connection, you lose your tunnel as well. But you only lose your tunnel, so if you started in a screen, it will continue to be running on the Jupyter notebook, but your tunnel will die. So it's very easy to quickly reload it by just opening your tunnel again, and you don't have to rerun the Jupyter notebook command on TSCC.
If you open it initially in the screen.
If you open it initially in the screen. Were you guys able to try this? Did anybody make a screen, go into it, go back out, go back into it again? Okay. They're really useful. I use them a lot. If you want to get super fancy, you can even have multiple tabs within a screen. Okay, so here's an example I can show you. I'm on an interactive job here, and you can see it looks a little bit different because I've got some sort of job number here. So here I'm operating on an interactive job. If I do Well, I just killed my screens. But within screens, you can even have multiple tabs open. And if you start to do more advanced stuff, you can Google more about that. Okay. You can. This is why screens can be a little bit confusing. Sometimes you mess things up. Anytime you're in a screen and you want to kill it, you can just type exit. And if you just keep typing exit, or Control AD will get you out of the screen, and then you can see if you have any of them running with screen-R. You can also just keep typing exit until it takes you back to your head node and logs you out of TSCC. Yeah.
I don't know if you said this before, but is it advantageous just to always set your wall time to as largest as the interactive jobs because it might die on you right when you're in the middle of something?
Yeah, it depends what you're doing. So and it also depends on the queue. So some of the queues have max limits. So the hotel queue has a max limit of an 8-hour wall time. Because we run, in the YoLab, our standard is to run the Python notebooks on interactive jobs. There's some people in my lab that, and because we have our own queue, we can run it for however long we want. They will start an interactive job that lasts a week, so however many hours that is, and we'll just continue to run their notebooks for the full week on there. I typically run my interactive jobs for like 8 to 14 hours, hoping that I'll be able to get through the full workday without it dying on me. But keep in mind that you're using memory, and it costs money anytime you're using stuff from TSCC. So be wise about it. Don't reserve it for a week if you don't need it for a week. But if you want to be using your interactive job for all the processing that you're going to be doing that day, it's a good idea to overestimate a little bit. You can bump it up to maybe 16 hours if you have access to a queue that goes that long, and then do all your processing in that. Yep. Yeah, so the train accounts, I'm not sure how long Jim keeps those up, but I mentioned in the beginning, and there's links to the TSCC website. The other thing that they offer is a 90-day free trial for your own username on TSCC. So you're not going to have all the installations that you've done already with this class, but you have access to all of the notebooks with all of the instructions in there, so you should be able to recreate anything that you want. So you can do a 90-day trial with TSCC for free, and then depending on what lab you guys are in or will be joining, that's something you can talk to your PI about. I'm not sure about the cost, and I'm not sure how it works for individual users within labs, but Jim is really helpful, and you can feel free to email him to get more information about how you would actually get accounts. Okay, let's take a quick 5-minute break. I have one more activity that we're going to do using bed tools. Bed tools is used all the time in bioinformatics, so I want to introduce you guys to it, but I'm going to do it in a way that is kind of more of a group exercise and less me teaching and telling you guys what to do. So we'll get to that after the break. And again, remember, today we're finishing up at noon, so there'll be lunch at noon, so we got about an hour and a half left. So 5-minute break, and then we'll get into the exercise, and you guys will work in small groups for that one.